This is an opportunity to celebrate the incredible department that we have. As you know, we have over 700 people on faculty. You know, we're the second largest department, and our research budget has increased by 18% in the past year. So we're doing great, but we often don't know everything that is going on. So this is an opportunity to do that. Here's the order of presentations. So we're going to start with Richard. Okay, go.
All right, thank you, Maurizio, and good morning, everyone. So the focus of my research is on self-interest thoughts and behaviors and depression, and it's suicide and depression. And the primary focus in terms of population of my work is with adolescents, with a little bit of spread to young adults and pre-adolescent children. And the bulk of my work is really to focus on trying to understand mechanisms and processes of risk, with a focus on state-sensitive and temporally delimited risk factors. And the reason for this is that they might be the most promising candidates for short-term risk factors. Through work in this area, my eventual goal is to help advance our ability to predict these clinical outcomes, particularly suicidal behavior, and therefore to prevent them. Can we go on to the next slide, please? All right, so I'm going to describe very, very briefly two active grants I have in this area. The first grant aims to see if we can reasonably accurately detect or predict, I should say, risk for suicide attempts in a sample of adolescents psychiatric inpatients within 30 days after discharge from inpatient care, which is a particularly high-risk period for these teens in terms of risk for suicidal behavior. And the focus here is on looking at a broad variety of neurocognitive measures administered with the teens while they're on the unit. The second grant here aims to see if we can identify windows of acute risk during the 30 days after discharge through continuous monitoring of life stress. So the translational aims of the grants are to look at, to be really complementary. The first one looking at a snapshot of risk so that it could be used in places where we only have one-time assessments, such as inpatient care, ED settings, when we have to make a single clinical decision of risk. And the second grant is aimed at translationally at continuous monitoring of risk where settings where we have repeat assessments, such as outpatient care.
Thank you.
Right.
Thank you, Richard. All right, thank you so much. Great job. Thank you. Next, Division of Neuropsychiatry and Neuromodulation.
Thank you, Maurizio. So I'm here to present on behalf of the division. We're a group of about 50 people, working clinicians, patient scientists. We have about almost 20 faculty, 6 key awardees, and our work both clinically and research really focus on human circuit neuroscience. We try to understand the anatomy, the physiology, and the behavioral computations of brain circuits. To do that, we use 4 main types of methods. We use brain stimulation, the whole gamut, to probe the brain. We use neuroimaging and neurophysiology to measure brain activity, brain anatomy, and physiology. We use behavioral tools, and we use clinical assessments. And the scope of our research is really spans from understanding basic human neuroscience to understand mechanism of disease, pathophysiology, the circuit level, and the core is really on translation and trying to identify and develop biomarkers and predictors to target discovery and eventually develop novel treatments. Next slide. So one of the examples is this study by which we try to leverage the fact that ECT that we generally think of is very non-specific brain stimulation technique. We know a lot about it clinically, and we know that it's very effective, it's rapidly acting, and it has innate anti-suicidal effects. So we try to use ECT combined with imaging and behavioral assessments to understand how does ECT change the brain in order to lead to these anti-suicidal properties. And we focus on this area you see on the left, the rostral anterior cingulate, because it's an area and lots of evidence from postmortem studies, metabolic PET, structural MRI, functional MRI have pointed that carries a signal of pathological relevance in suicidal patients. So we ask the questions, is the connectivity between the anterior cingulate to the whole brain before ECT predict who will do better, meaning who will reduce the suicide risk and not? And the second question is, would the change of the connectivity between the ACC and the whole brain explain how suicidal risk is diminished? And indeed, we found that the connectivity between this node and the right inferior parietal lobe both predicts and explains the connectivity, the risk of suicidal risk. So we have a circuit level biomarker that is both predictive biomarker and a response biomarker for suicidal risk, for suicidal anti-suicidal properties. And the question is now, could we use other means, other techniques that are less invasive, that are more focal, that can be more personalized to induce these very same changes that we see ECT is inducing in order to reduce suicidal risk. So to do that now, we have a study that is using accelerated neuroimaging guided TMS in order to induce these very same changes, induce these changes between the connectivity of the right IPL.
Joan, it's already 2 minutes, so sorry meant to cut you off. Thank you.
That was it, yeah, thanks.
Hi, my name is Taylor Burke, and I'm a clinical psychologist within the Division of Child and Adolescent Psychiatry and the Center for Precision Psychiatry, and I joined the faculty this past August. My research aims to use novel methodologies and computational approaches to improve our ability to identify youth at short-term risk for suicidal and non-suicidal self-injury. Next slide, please. And so I wanted to highlight two of my recently funded studies. My K23's overarching goal is to assess whether acute behavioral changes from an individual's typical patterns of social engagement, sleep, and physical activity indicate proximal risk for increases in suicide risk. We'll be using mobile sensing, actigraphy, and idiographic modeling for this aim. And the study further aims to characterize the intra-individual network structure of these behavioral factors and suicidal ideation to enhance suicide risk assessment and inform future intervention. And so then in my R21 and AFSP funded study, my goal is to leverage computer vision techniques to automate the assessment of hypothesized self-injury visual severity indicators, and then determine the utility of these visual signals in predicting suicide risk. And my hope is that this proof of concept study might set the stage to pursue my long-term goal of integrating this technology into psychiatric care entry points to assess whether it can serve as a clinical decision support tool.
Terrific. Thank you so much, Taylor. You did less in 2 minutes. Next, the BMed program, Connell.
Good morning, folks. It's really our postdoctoral fellows and our K awardees who are really leading the charge in behavioural medicine this year. Dr. Calvin Fitch is awaiting his Notice of Grant Award for his K to develop an innovative M-Health intervention to address the mental health and structural barriers experienced by young sexual minority Black men. Dr. Amelia Stolton is also awaiting her Notice of Grant Award for her K to develop a behavioural intervention to address the mental health barriers to PrEP uptake among pregnant and postpartum women in the townships outside Cape Town. Dr. Abby Batchelder, although in the penultimate year of her K, has been awarded 4 grants, including most recently an R34, to extend her work on stigma-informed treatment for substance use. Dr. Peter Chai, also in the penultimate year of his K, has just been awarded the prestigious Avenir Award and multiple other grants in the past year to extend his groundbreaking work to develop the technology and application of the digital pill, which I don't have time to explain here. We are lucky enough to work with some of the most talented CRCs around. One of these extraordinary folks in our group, Brett Dolatona, last month published a first author commentary in JAMA Network Open, a monograph on improving mental health among transgender and gender diverse youth. Check your rearview mirror, folks. There are some very talented researchers coming up fast. Next slide. As our senior members of behavioural medicine plug away at our multiple domestic and international clinical trials, thank you, Dr. Christina Soros, we are proud of 2 significant extensions of our work poised to make a real impact. Firstly, we've established a strategic alliance with the City of Cape Town Department of Health through a joint implementation science partnership to extend our mental health treatment supporting HIV across the townships. And we've been invited to join the Ministry of Health Task Force in Zimbabwe that's updating the national HIV treatment guidelines. Both pathways to get empirically supported mental health treatment into the resource-limited settings where they're needed. Thank you very much.
Thank you so much, Connell. Next, Alex. Alex.
Thanks. Our central goal in the program on social behavioral health is to explicate the biological and behavioral mechanisms through which large-scale social forces like stigma and discrimination exert adverse influence on population mental health. We have a number of RO1-funded studies in Uganda and South Africa to understand how HIV stigma and discrimination worsen mental health and HIV treatment outcomes among people with HIV. We also have a domestic research program to understand how structural racism, principally manifested through racially discriminatory policing, worsens mental health and sleep health among Black people in the US. Next slide, please. Earlier this year, we published a paper in Nature Human Behavior that emerged from one of our R21 studies in Soweto. For those of you who might not be familiar with this field, the theory of syndemics comes to us from medical anthropology, but a lot of the evidence supporting this theory is derived either from ethnographic studies or from epidemiological studies that have a lot of limitations. To be honest, I'm not really sure how or why this paper landed in Nature Human Behavior and with an editorial, which was doubly surprising. Probably the most novel thing about our study was its unique pairing of ethnographic and population-based research to triangulate and characterize a novel syndemic of communicable and non-communicable disease in this setting, which has not yet been done before. Our group is actively welcoming new investigators. A new hire from Northwestern, Ronke Peterson, just got a fundable score on a K-23 application to study and intervene upon mental illness among Black people in the US. So if you see her around, please say hi.
Thank you so much, Alex. Next, Carmel Choi.
Hi, everyone. It's an honor to be part of the lineup this morning and to hear about such inspiring labs and programs in our department. So I'm a clinical psychologist and the director of the Precision Prevention Program in the MGH Center for Precision Psychiatry. And this program is focused on clinically informed, data-driven, and life course approaches to 3 complementary research themes. 1, understanding the nature of psychological resilience, including how genetic and environmental factors shape resilience across the life course. 2, identifying modifiable protective factors for mental health, especially behavioral, social, and environmental factors that robustly protect against depression and PTSD. And 3, predicting the real-world risk of stress-related conditions using large-scale data from health systems, et cetera, to identify those who could benefit from resilience promotion efforts. Next slide, please. So some recent work from each of these 3 areas is shown here on the left, a study of resilience where we showed that among older adults who experienced a major life stressor like divorce or illness, we could combine genetic risk scores for psychiatric and health traits using machine learning approaches to accurately differentiate those who showed a chronic depression trajectory versus those who showed recovering or resilient trajectories. Now in the middle, a study in AJP where we conducted an exposure-wide and Mendelian randomization analysis of over 100 modifiable factors with incident depression in the UK Biobank, which highlighted the potential causal importance of social connection, sedentary media use, daytime sleep, and previously exercise for depression in adults. And we've been extending this work now to younger individuals where primary prevention may be most possible. And this feeds into the last area on the right that's funded by my K08, focused on stratifying adolescent depression risks or SAD risk using electronic health records. And so I'm excited to share more about this soon, and I'm looking forward to collaborating and continue to learn from many of you along the way.
Thank you so much, Caramel. Next, the HPRRR.
Thank you. We pronounce it HIPPR, and I'm thrilled to give a brief overview. We're about to hit our year anniversary, and the intention of this program was to combine interdisciplinary behavioral health services research. Specifically, our overarching goal is really to integrate behavioral and psychosocial health into the medical treatment of individuals and communities. We want to enhance individuals' health, wellness, and resiliency, and really figure out how to integrate these innovative treatments and models of care delivery across hospital and community-based systems. Overall, our investigators conduct what we're focusing on behavioral science delivery research, and we talk about healthcare access and utilization. We focus on mind-body interventions adapted for different patient and community-based populations. We're building bridges between oncology and primary care, a real focus on treatments that are accessible and sustainable, and also with the lens of health equity and health justice in terms of our research and the researchers that we're developing. We're leveraging innovative technology-based means of intervention delivery, emphasizing community outreach and partnerships in our work, and executing externally funded research and implementation of behavioral treatments. Next slide, please. So overall, this is the HIPAA investigations. They fall into the buckets of survivorship, prevention, resiliency, and integrative medicine, health equity, and impact of COVID. We have a rigorous program developing clinical researchers, starting with college students, going up through CRCs, junior faculty across psychiatry and medicine. And I don't have time to go into each of these, but these are just the areas of the HIPAA investigators. We've had a diversity of funding through NIH, the American Cancer Society, HRSA, and foundation grants, and the National Comprehensive Cancer Network. And I'm at 2 minutes, so thank you very much.
Thank you so much, Elise. Next, Daphne.
Oh, okay, great. Yeah, nice to see everyone. So I'm just going to talk about the lab that I run in Charlestown, the Emotion and Social Neuroscience Lab, which, we use cognitive neuroscience and behavioral methods and neuroimaging, including high field imaging, to study emotion and social cognition, because these processes have been linked with some of the symptoms of serious mental illness like schizophrenia. And we also study these processes in at-risk groups. And, our overarching goal is really to identify markers, sort of the underlying mechanisms that we can target. And so in recent years, we also have this kind of related program, the Resilience and Prevention Program called RAP, where we try and translate these findings to develop and study behavioral interventions that are delivered via a number of different modalities, in person, Zoom, and more recently using virtual reality, where we are sort of aiming to reduce risk and improve outcomes in people who are at risk for serious mental illness. Next slide, please. And I'm also obviously co-director of the schizophrenia program, which I'm not going to talk about today. So I'm just going to mention this one finding that we've had recently that's received a fair amount of attention in the media for some reason, I guess because of its pandemic relevance. So we've done a number of studies, as I mentioned, using virtual reality methods. And so we were studying personal space. We've been studying personal space as a dimensional marker of social functioning before the pandemic, using both conventional methods and virtual reality. And we found that during the pandemic, the size of personal space, so the distance where you feel comfortable sort of maintaining from other people, into which other people can't intrude without eliciting an arousal response, that size, that personal space boundary has expanded during the pandemic. But the interesting thing, in some ways that's not surprising, and we saw that it correlated with social distancing behavior, but you can see that it was an equal increase both to humans, in response to humans, and also avatars, suggesting that some really basic automatic brain mechanism has been altered by the pandemic. And so we're studying this further using high field imaging, 7T fMRI, and also we have a behavioral intervention using multiplayer immersive VR to try and reverse this abnormality. So we'll see.
Thank you. Thank you, Daphne.
All right. Thank you.
Next, Andy.
Great. We have a very broad portfolio in the Dalton Center, but I just wanted to highlight 2 studies before I pass the baton on to Louisa. These were 2 large PCORI studies. The first one looked at 3-session online versus 8-session online mindfulness-based cognitive therapy, and we found no difference between the 2, which was a big surprise. And then we also did a study for people who had mood disorders and cardiovascular risk. And we looked at a Fitbit alone versus a Fitbit plus cognitive behavioral therapy and a Fitbit plus mindfulness-based therapy. And we looked at the number of steps that people took. We actually sent out over 500 Fitbits and got 312 that were usable. And mostly we found that people didn't change that much, which was really the bottom line with that. And now I'll pass it over to Louisa.
Great, next slide. Thank you, Andy. Good morning, everybody. As Andy said, we have many projects going on, but we wanted to talk about one sort of clinical innovation because this might be relevant to many of you on this call, especially who see and treat individuals with bipolar disorder. So this is a new clinic launched within the Daughton Center, or the Focused Integrated Team-Based Treatment for Bipolar Disorder. This is not sort of your standard clinic where we're seeing people every week for therapy. This is a stepped care model where individuals will get asynchronous and or group support and or support from peer specialists, et cetera, before even getting to level 3, which would be, or rather level 4, which is getting to sort of individual therapy. So we're really excited about this. It's a data-driven model or learning healthcare system. So we're tracking individuals very carefully to learn how they do with each intervention that we do. So we're really excited about it. And if you have questions about it, please let us know. Thanks.
Thank you. Okay, the Sahe Lab.
Yeah, good morning. The goal of my lab is to improve hippocampal dependent functions and cognition. We spend a fair bit of time thinking about circuits and cell types within circuits and wiring diagrams of the hippocampus with the goal of identifying genes that prescribe connectivity, and that might be harnessed to re-engineer the wiring diagram in diseased brains. We also spend a fair bit of time thinking about genes that prescribe changes in non-neuronal cell types like glia, microglia, that we know become inflamed in many different brain diseases, and neuroinflammation is a risk factor for many brain diseases as well. The ultimate goal is to harness these genes and build a pipeline that allows us to target these genes using gene therapy modalities, including AAV, ASO, siRNA, to improve cognition. One example, the next slide, is a gene that we recently identified, and this illustrates our approach. We found that this gene dials up GABAergic inhibition in the brain. We know that GABAergic inhibition is disrupted in many different brain diseases, from autism to epilepsies to mild cognitive impairment, early stages of AD. And what we found is that targeting this gene allows us to restore GABAergic inhibition in the brain. And we have a patent now, and we're now optimizing the delivery modality and de-risking this asset. And this is true for other assets in the lab as well. And the goal is to ultimately achieve broad therapeutic impact across clinical diagnoses that classify these disorders as very distinct, when in fact they all exist along the continuum. So stay tuned. Thanks.
So my today's talk will focus on one of our recent publications entitled Manipulating Placebo Analgesia and Nocebo Hyperalgesia by Changing Brain Excitability. So placebo and nocebo effect play an important role in medical practice. This study aims to investigate if we can modulate placebo and nocebo effect using non-invasive brain modulation method we call the TDCs, transcranial direct current stimulation. Healthy subjects were recruited and randomized to one of the 3 groups. We have a nodal TDCs, cathodal TDCs, and the sham TDCs. In this study, we used a well-tested expectancy manipulation model and experimental heat pain applied on the forearm to produce placebo analgesia effect and the nocebo hyperalgesia effect. Next slide, please. So we found that compared to sham, cathodal TDCs can enhance the placebo analgesia. Brain imaging data analysis showed cathodal TDCs can also increase the brain activity at medial prefrontal cortex, a key region involved in placebo analgesia based on the literature, and also can enhance the connectivity between the dorsolateral prefrontal cortex, which is the target of TDCs, and this medial prefrontal cortex. In addition, we also found that a nodal TDCs can reduce the nocebo effect and decrease the activity of insular, a key region that believed to be involved to encoding the pain intensity, and also decrease the connectivity between the DLPFC and the insular during the heat pin application. So in summary, our findings suggest that changing the excitability of the dorsolateral prefrontal cortex using TDCAs can modulate placebo and nocebo effects, which may have the potential to improve clinical outcomes in medicine. Thank you for your attention.
Thank you, Gian and Randy. Next, systems consolidation of memory.
Okay, this is actually Dara, and good morning, everyone. There's a wealth of evidence that sleep plays an evolutionarily conserved role in memory consolidation. After learning, memory is not just stabilized like a snapshot, it is enhanced and transformed in the brain so it can guide future behavior. And in this way, memory is more prospective than retrospective. Memory consolidation depends on the orchestration of 3 brain rhythms during sleep. Cortical slow oscillations group thalamic spindles in their excitable upstates, and memories are replayed in the hippocampus during ripples, which nest in the troughs of spindles. Next slide, please. It is this interregional dialogue that transfers selected new memories from temporary representation in the hippocampus to longer-term storage in the cortex. My lab studies how this dialogue breaks down in neurodevelopmental disorders such as schizophrenia and autism. In schizophrenia, we have shown impaired memory consolidation that correlates with reduced spindle activity and spindle slow oscillation coupling. And children with autism also have spindle deficits and a breakdown of this coupling. We initially tried to improve memory with the drug azopiclone, but were not successful. And this probably reflects that although it increased spindles, the azopiclone also messed up spindle slow oscillation coupling and suppressed hippocampal ripples. We are now testing the effects of non-invasive brain stimulation using closed-loop auditory stimulation or CLAS on all 3 oscillations and their coupling. Wearable EEG headbands are user-friendly and can both characterize sleep physiology and also deliver CLAS at home. We hope to turn this closed-loop auditory stimulation into a safe, scalable, home-based intervention to enhance the coordination within this thalamocortical hippocampal network and improve function in a range of disorders. Thanks for your attention.
The Sleep and Anxiety Disorders Lab, the goal is to study how poor sleep contributes to mood and anxiety disorders, and in particular to PTSD. Following an emotionally traumatic event, most individuals fully recover. However, there is 7 to 9% who develop PTSD, and we're interested in what may determine who does go on to PTSD. Fear extinction memory, that is, remembering that a trauma reminder no longer signals danger, has been suggested to be deficient in PTSD. Sleep promotes memory consolidation, thus poor sleep may interfere with the consolidation of extinction memory. There is much evidence now that poor sleep contributes to the etiology of mood and anxiety disorders, including PTSD. Recent fMRI findings from our laboratory suggest one way in which this may occur. We've conducted a fear conditioning and extinction protocol while scanning with fMRI. And we compared 3 groups, one with primary insomnia, another healthy controls that were sleep deprived, and a third with PTSD to their respective control groups. In all 3, we observed a similar pattern. The control groups, in this case, you see the trauma exposed without PTSD and the fully rested, were all activated prefrontal regulatory extinction-related structures during extinction learning that was immediately done following fear conditioning. However, in the pathological groups, there you see PTSD, or in the case of the model of normal adults, the sleep-deprived group, the engagement of these same areas was delayed for 12 to 24 hours, depending on the study. So next slide, please. This illustrates that extinction being a new memory is competing with the previously instilled fear memory. And what we see also in the healthy individuals, the ones without insomnia, sleep deprivation, or PTSD, is that they activate both the executive areas of the prefrontal cortex as well as areas of the salience network. In this case, we see during extinction learning, the good sleepers all activated both prefrontal and salience structures, whereas the insomnia folks did not. Following an actual trauma, delayed engagement of extinction leaves the clinical or the sleep-deprived individuals vulnerable to additional traumatization or to developing excessively fearful attributions.
Thank you, Ed. Next, Yakel.
Hi, good morning, everybody. So I'm Yakel Quiroz. I'm the director of MAP, or the Multicultural Alzheimer's Prevention Program. And at MAP, we follow a cultural approach to dementia research and dementia prevention. So we start by including ethno-racially diverse patients and family in all of our studies. We leverage our expertise in multicultural neuropsychology to use cultural informed assessment. We also work on the genetic characterization of individuals at risk for Alzheimer's disease and other dementia. We use a multimodal neuroimaging approach and biomarkers for early detection and prediction of risk. And we are working now our way to use all of this to inform the interventions and clinical trials. Next. So the four main projects that we have right now, and they're in the middle. So the main project is the Columbia-Boston Autosomal Dominant Alzheimer's Disease, or COLBOS. The one that we just launched last year is another one that is the Boston Latino Aging Study, when we follow individuals that are older than 55, and it's a biomarker study. So if you have any older Latinos, please refer to us. The one that we just launched a few months ago is the Healthy Aging and Resilient Brain Study, when we are following individuals that are resilient, that are in the oldest all, that are from diverse or underrepresented groups. And the one that we have now over a year following is the bilingual individuals that are bilingual with mild cognitive impairment and dementia. Also, if you happen to have any of those patients, please refer it to us. Our main goal is to actually advance the research in biomarkers and protected at risk factors for age-related cognitive decline and dementia, with a specific focus on multicultural populations. We have several collaborations, and here I'm only including the ones that are relevant to the MGH and MGH community. We have sub-studies looking at genetic factors, eye biomarkers, sex differences, sleep, physical activity, other biomarkers, cardiovascular factors, cognitive markers, chronic stress, and of course, tau biomarkers. And always open to more collaborations. So please reach out if you have any interest in working with me. Thank you.
Thank you so much, Yakel. Next, the MGH Brain Photo Biomodulation Program, Paulo.
Thank you, Maurizio. So I'm presenting today on the MGH Photo Biomodulation team. My name is Paolo Cassano. I work within the Division of Neuropsychiatry with Joan Camprodon. First of all, what is infrared light? Infrared light is natural. 50% of sunlight at ground level is infrared light. And we are shedding the light transcranially. However, it's not invasive. It's really LED over your head, LED or laser-based light. It's safer, it's safe, and also well tolerated. We have several studies. We're treating Alzheimer's disease, mild cognitive impairment, depression. We're treating Down syndrome and bipolar disorder. We're also collaborating with other groups. For instance, child psychiatry with Attila Soranoglu and Maya Uchida are running their own studies, and as well as the bipolar group with Andy Nirenberg and Emmy Peters. So if you're interested in doing these studies, please reach out. We're eager to collaborate with you and to also consider other applications. as well as if you have patients who have, for instance, mild cognitive impairment or mild dementia, who are over 65, please refer. That's another way for us to collaborate on that. So next. So this is our study that we conducted together with NYU. We showed that there is an effect of infrared light on ball signal. Ball signal is basically depending on oxygen consumption and oxygen flow. Here on the left, you see that the red high dose had no effect on ball signal. The low dose decreased the ball signal. The sham had no effect, and the medium dose increased the ball signal. So we see here a dose-dependent effect and target engagement with this treatment. Therefore, this is very promising. We are bringing this to the next level to find efficacy trials. Please feel free to reach out and collaborate. Thank you.
Thank you, Paulo.
It's a pleasure to update you on the clinical research portfolio of the Amin Pinizzotto Center for Women's Mental Health. Our research mission really is to enhance the understanding of factors that increase the risk for reproductive associated psychiatric disorders manifesting across the lives of women, whether that be during pregnancy, the postpartum period, or later in life in women who are peri or postmenopausal. We are interested in investigating the pharmacologic and non-pharmacologic interventions that mitigate suffering for women at such critical times in their lives. We have a broad portfolio of clinical research activities, and I've listed them here, and just a sampling of everything from a study to see how we can prevent relapse of depression during pregnancy and the postpartum period, to using digital applications to treat perinatal depression, to evaluating the reproductive safety of some of the medications that are used by reproductive age women during pregnancy and the postpartum period. And we're very interested in evaluating the most efficacious therapies to treat reproductive associated mood disturbance, whether that be in pregnant or postpartum or in peri or postmenopausal women. The MGH Postpartum Psychosis Project is sort of a specific example of our work. This is a study which we designed to delineate the phenomenology of postpartum psychosis, one of the most severe forms of postpartum psychiatric illness, with an effort to identify both the genomic and clinical factors that increase risk for postpartum psychosis. We have been very successful in engaging one of the largest cohorts in the world to date of women who have suffered historically of postpartum psychosis. And then, aside from very deep phenotyping of these subjects, we have partnered with Dr. Holt's lab to see if there are neuroimaging correlates that go along with postpartum psychosis. And this dovetails with the work of the Holt lab in understanding the neuroanatomic and functional underpinnings of psychotic illness in and of itself, looking at certain areas of the brain which may be implicated in psychotic illness. We have extended or have tried to leverage that methodology in P3 to see how that might be applied to women who have suffered from postpartum psychosis. Hey, thanks so much.
Jill.
Hi, thank you for including me in this. It's just a wonderful idea, Maurizio, and I've learned a lot from what's going on in the department that I didn't know. So we're the Clinical Neuroscience Laboratory for Sex Differences in the Brain. We're part of the Innovation Center on Sex Differences in Medicine. We enhance discoveries around sex differences and risks for disorders of the brain and their co-occurrence with cardiometabolic disease. We have an interdisciplinary team. We work with multimodal imaging, hormones, immune pathways, cardiac physiology, genetics, and genomics. We take a lifespan approach, that is when the brain and the body are either differentially flooded or depleted of gonadal hormones, beginning in fetal development and ending with aging and menopause. The circuitries we focus on are the stress circuitry, which also regulates mood and anxiety, and memory circuitries. And we develop novel therapeutics that are targeted for depression and its co-occurrence with cardiovascular disease and also Alzheimer's disease. Next slide, please. We've had some really cool findings in the last year. So I'm going to whip through them, but if people are interested, please let me know. We're really interested and study for many, many years now prenatal stress models of sex differences in the risk for major depression and its co-occurrence with loss of cardiac tone, loss of parasympathetic regulation of the heart, which is a major risk factor for cardiovascular disease itself. And we're interested in part through the impact of immunity and the hormonal regulation of that. And we use high-risk pregnancies as a model to look at these prenatal stress/immune models. And in this case, and we've been following a prenatal cohort who are now turning into their 60s. In this case, we found in a recent paper, in previous work, we found that changes in the maternal prenatal pro-inflammatory cytokines during really due to some of these high-risk pregnancies, like preeclampsia, were risk factors, sex differences in risk for major depression and loss of cardiac tone. And in this case, we found the mediating effects were sex-dependent effects on the stress circuitry in the offspring and the dysregulation of the hippocampal control of arousal on hypothalamus. And so one of the things we also found with this circuitry also affected and dysregulated autonomic nervous system dysregulation, and we're developing a respiratory gated transcutaneous auricular vagus nerve stimulation device that is targeted to depression, hopefully also in its co-occurrence with hypertension. We had a paper come out where we showed lower depressive symptomatology, lowering anxiety, and raising parasympathetic regulation of the heart. So we're very excited about that. There were acute effects that had clinical significance. And finally, we've been really interested in some of the sex differences in genes. And we had a recent paper come out using the Psychiatry Genetics Consortium data in which we looked at sex by genotype interactions and found 2 of the SNPs, significant SNPs, were in immune pathways. And the last thing I want to say is please come visit us at our website. If you're interested in this, please come join us at the Innovation Center if you are interested in collaborating. And I want to give credit to a few different people in the lab. Junior faculty, Ronald Garcia, Kyoko Konishi, research scientist Justine Cohen. So
thank you, Jill. Alex, I think it's Alex Lucier or Erin. Erin?
Morning, everyone. My name is Alex Lucier. I'm a postdoctoral research fellow in Erin Dunn's lab. Very pleased to be presenting these slides on her behalf this morning. The goal of the Dunn Lab broadly is to identify strategies to prevent depression as early on in the lifespan as possible. And we do that through 4 main pillars. The first is identifying who is at higher risk of depression using genetic and other markers of vulnerability. The second is to understand how mechanisms of childhood adversity get under the skin. And then our third goal is to determine when in the lifespan experiences, including adversity, matter the most. And finally, we're building new infrastructures for scientific knowledge translation, including through the Science of Adversity and Resilience Initiative. Next slide, please. As an example of some of the really exciting work coming out of the Dunn Lab these days, last November, we published this paper in JAMA Network Open, which was really the first study of its kind to examine the association between prenatal stressors and this stress line in teeth called the neonatal line. The neonatal line is thought to reflect more stressful births, and so we examined it in the context of several psychosocial experiences. Although we found no associations with neighborhood level factors, we found that children born to mothers who reported higher psych pathology during pregnancy and across the life course generally had wider neonatal lines suggestive of higher stress. By contrast, social support, which is generally thought to be positive, resulted in thinner neonatal lines. Now we're currently working to replicate and expand this study through the STRONG study, which is a study of children born to mothers who were pregnant or had newborns at the time of the Boston Marathon bombings. The goal of this work is eventually to use baby teeth as a supplement to biomarkers to identify those at highest risk for depression. Thank you for your time.
Thank you, Alex. Next.
That's me. Good morning, everybody. So nice to be here today and to hear what everybody's doing. So what I would like to do is today is like just illustrate what I've been working on over the past 5 years. So over the past 5 years, I've been interested in addressing several gaps present in the field of psychiatry that might have been preventing important advancement in treatment of patients. So I see 3 main areas of challenges in our field. So first of all, we lack an objective test to detect the presence of a psychiatric diagnosis or to measure symptom severity. So in psychiatry, unfortunately, we do not have a blood test or device that could easily objectively measure the presence of a psychiatric disorder or its severity. Secondly, we do not know when patients are acutely in need of assistance, and we do not have tools to provide help in real time. And then finally, for many decades, we've been seeing research studies that focus on developing treatment derived from population-based data, missing important differences present within individuals. So I've been focused on the 3 main areas of studies. Next slide. So at the DCRP, we're conducting a number of studies, but today I'm presenting one specific finding that was published in the couple of years ago, last year. So during this study illustrate results from an 8-week-long study where participants were wearing daily 2 wristband watches that measure physiological indices, and then they downloaded an app on their phone that monitored socialization and activity levels. And in the figures here is illustrated the accuracy of the models that were created extracting data from either the mobile alone or the wearable, or combining features from both wearables and mobile sensors. So I've been very interested in identifying like how much data we need to identify severity of depression. And from this figure, we can see that the time split model, and just to say a little bit more about the time split. So we basically ran machine learning and we divided the data using the first 3 depression measure to predict how they were doing later in the study. And we saw that that model was actually more accurate, and the model using data only from the wearable was again more accurate. We used so lower number just to illustrate this with people that are not familiar with this kind of data analysis. Lower data is increasing, lower numbers depict higher accuracy. So the data again from the model from with the data from the mobile was the most accurate. So my hope is that with this line of study, we can see that digital health, so sensors can be leveraged to improve our ability to detect depression, to also know when we need to intervene, to develop just-in-time intervention, and to also develop more personalized treatment.
Thank you, Paula. Appreciate it, Chris.
Thank you so much for inviting me. Just very briefly, a little bit of history. We've been conducting research on ketamine and treatment-resistant depression since 2014. And this led to, in 2018, to open the intravenous ketamine clinic for depression at Mass General. The access has progressively increased. Currently, we have 60 slots per week, and we have almost half of the patients who are covered by insurance, giving also some access to inpatients. Soon we will open an esketamine clinic, which is FDA approved and will be covered by most insurance. On to the research, we recently obtained a large AFSP grant where we will offer ketamine maintenance for patients admitted for suicidal ideation or suicide attempt. So the treatment for 6 months will be covered through the study with the goal of preventing reattempt of suicide or rehospitalization. And we have a collaboration with Atai looking at MRI and EEG biomarker of neuroplasticity and structural changes with ketamine as a possible model of mechanism of action with psychedelic. Also, we have submitted a possible a grant for investigating the longitudinal changes on MRI for patients who have been on ketamine maintenance for over 2 years. And another study to investigate the mechanism of action of ketamine with a double PET ligand, dopaminergic and glutamatergic receptor ligand. And this is submitted and under review. Thank you very much.
Thank you, Chris. Perfect. Center for Addiction Medicine, Jolie.
Hi, I will make this very quick. We have a lot going on at the Center for Addiction Medicine. I'll give an overview of some of the neuroscience studies, and then Randy Schuster, the next speaker, will talk about some other studies that we're doing. But basically, our goal at the Center for Addiction Medicine is to understand how cannabis and other substances affect the brain and clinical outcomes. So I'm interested in neuroscience, and we have studies of functional MRI, PET, FNIRS, and so forth to understand this phenomenon. We have several new studies going on this year. We're looking at medical cannabis and prescription opioid taper support for reduction of pain and opioid doses in adults with chronic non-cancer pain, and that's a recently funded R01 clinical trial. We're also looking at cannabidiol or CBD for reduction of brain neuroinflammation using PET scans. We also got a secondary research analysis grant to look at the adolescent brain and cognitive development study, or the ABCD study, which is just a massive undertaking by the NIH to scan 10,000 kids every 2 years for 10 years. Next slide, please. So I'm just going to briefly present a snapshot of some of our recent findings, which we're very excited about. We recently published a paper in JAMA Network Open, which was a result of a clinical trial of medical marijuana cards in patients seeking medical marijuana for various conditions. And the punchline was that it was effective for self-reported sleep, which is a finding I'd like to follow up with objective sleep measures, but it was not very effective for anxiety, depression, or pain. The next study was sort of a subsidiary study where we looked at the urine of these patients using medical marijuana and found that there were no detectable cannabinoids in a lot of the urine. So sort of buyer beware. And then finally, we just published a finding looking at using FNIRS to look at intoxication in the brain, which was very exciting. So I will leave it at that.
Thank you. Thank you, Jody. Next, Randy.
Hi, good morning, and thanks so much for the opportunity to highlight some of my work. So I'm a clinical psychologist at the Center for Addiction Medicine with Jody and with Eden Evans, and we have a really incredible multidisciplinary group of psychologists, psychiatrists, neuroscientists with a very active and diverse portfolio of research. So at CAM, my work has adolescent substance use, namely cannabis and tobacco use, as well as community-based approaches really at the core of its focus. We have several active grants that I'm leading or co-leading that currently span really 2 thematic areas. So first, we have several grants focused on developing and testing novel interventions for adolescent substance use. So one project, which has been funded for the past 3 years by the Massachusetts Department of Public Health, is one in which we developed a novel drug agnostic substance use diversion program, which we're calling I Decide, which aims to provide schools with a more equitable alternative to punishment for school-based substance use infractions. In January, we launched a very large pragmatic clinical effectiveness trial of this intervention in over 75 schools across the state. Another that I want to highlight is one that I'm co-leading with Eden Evans. So despite the epidemic of vaping and the rising number of kids reporting interest in quitting, as recently published in JAMA this month, there are no evidence-based treatment regimens to support cessation. So Eden and I are co-leading an R01 clinical trial funded by NIDA, in which we're aiming to test the efficacy of varenicline coupled with behavioral support for adolescent vaping cessation. A second camp of active grants are one in which we're aiming to evaluate changes in mood, cognition, and behavior with cannabis abstinence to better understand the residual effects of the drug in developmentally vulnerable populations. So for purposes of time, I'm not going to go through that. If we go to the next slide, just the one thing I'll mention here on the right, and something that we're quite proud of, is that all of our work is done in close collaboration with schools across the state. So we have a consortium that we worked really hard to develop over the past 5 years, involving over 100 schools that serve as active research sites. And I think this really sets the stage for a number of very exciting collaborations within the department. These serve as sites for both our clinical trials as well as longitudinal survey work. So we're well positioned to survey over 100,000 kids in the fall. So anyone in the group who has interest in big data and developmental populations, please reach out, and we welcome the collaborations. Thanks for the time.
Thank you so much, Randy. Next, Alicia.
Good morning. I direct the Laboratory for Youth Behavior, which is the research arm of Think Kids, a clinical program here in the psychiatry department. Think Kids itself trains and implements collaborative problem solving, an approach for treating children with behavior disorders and symptoms by building the cognitive skills children need to meet behavioral expectations. You can see the process here. Think Kids implements collaborative problem solving in large clinical systems and schools. Every provider or educator in the system starts using the approach. And when everyone is using collaborative problem solving, we see children and caregivers build specific competencies that lead to a number of positive outcomes. Next slide. In the laboratory for youth behavior, we study each step of that process and provide data, there you go, to help Think Kids continually refine the approach and the process to maximize benefits. So on one hand, we're intervention researchers. We study the outcomes and their mechanisms of the effects, and you can see examples of relevant research publications there on the right. And on the other hand, we're also implementation researchers studying the best ways to implement this complex approach in large systems. And we've published in implementation as well. We're always open to new collaborations. Thank you.
Thank you so much, Alicia. Next, Michael Murphy and Michael Jelinek.
Good morning. I want to begin with a little background about our MGH Pediatric Psychosocial Research Shop. Many years ago, our research interest took shape around a single problem, namely the recognition of mental health problems in pediatric primary care was very low, with only about 1% of all patients so recognized. And that although pediatricians routinely screen for medical conditions like anemia and diabetes, there was no standardized way for them to identify the 10% or so of children who are thought to have mental health problems. So our goals were to validate and disseminate a brief screen called the Pediatric Symptom Checklist, or PSC, to see if it could be used to improve mental health problem recognition. Since that time, we've written more than 40 papers using the PSC, and other investigators have written 250 more. Next slide, please. As of now, 30-something years later, we can report that we've reached these goals. Psychosocial screening is now recommended by the American Academy of Pediatrics for all children as a part of their annual physicals, and it's the standard of care here at MGH and in other large systems. And in fact, millions of PSCs are now completed each year. Our biggest accomplishment was the publication of an editorial in JAMA Pediatrics that summarized the evidence showing that screening was now being done routinely on a large scale in the real world and was identifying a higher percentage of children with problems. Our editorial went on to make the case that screening for psychosocial functioning should actually be considered as the 8th vital sign, as important to assess annually as pulse, blood pressure, BMI, and other vital signs, and strengthening the case for mental health as an essential component of overall child health, using the PSC as a tool to assess outcomes in our ongoing studies. Thank you.
Thank you, Michael. Next, it would be Robert Kincherf and Judy Edershen.
The Center for Law, Brain, and Behavior teaches the justice system about mental states. The law is all about mental states, but it uses an outdated 18th century model about human behavior and human brains. One example is that physical and mental injury, according to the law, are completely separate, or that substance use disorders are completely voluntary. Why do we want to change these notions? Because better decisions aligned with science cause better outcomes aligned with justice. And now I'm going to turn this over to Dr. Kincherf, our executive director.
Good day. I'm Robert Kincherf. I'm the executive director of the Center for Law, Brain, and Behavior. I'm a clinical and forensic psychologist and an attorney, and I'd like to share an example of the kind of work that we have been doing at the Center for Law, Brain, and Behavior. We recently submitted a white paper. This is a science paper focused on the science of late adolescence for a variety of legal issues. And this is just a brief timeline. We started in September 2020 and had a neuroscience summit, a multidisciplinary summit, which resulted in a period of work for over a year between October 2020 and December 2021. And it was released in January to, quite frankly, nationwide acclaim by judges, lawyers, juvenile justice reform advocates, law schools, policy centers, and others. The research relied upon has already been incorporated into 16 amicus briefs filed or joined by CLBB, including 6 Supreme Court cases. You can certainly take a look at the rest of that, but I just want to draw your attention to the number of states in which the white paper will be incorporated in appellate briefs and arguments in a number of states, and will be incorporated into international neurolaw training through the United Nations arm, a research and training arm of the United Nations. It has resulted in terrific scholarly productivity and is relevant to many legal and policy issues for late adolescents, ranging from the age of adult criminal prosecution to the nature of conditions of confinement. And it's impact on neurodevelopment and neurological functioning of persons. This white paper is available off of our website. It's an excellent model for the translational science that occurs when we try and take information from out of the laboratories and clinics and bring it into the courtrooms and legislatures of our country. Thank you for your time.
We have a broad range of studies in court, and Hillary will represent us with an example of a recently completed study. Hillary?
Thanks. Next slide, please. So this was a recent study where we aimed to get the best available data on remission rates after psychotherapy for body dysmorphic disorder to address a real lack of published BDD remission data. And this was a secondary analysis of Sabina Wilhelm and Kathy Phillips randomized controlled trial. So on the left, you can see our remission outcomes at post-treatment. So 68% of participants in CBT and 42% of participants in supportive psychotherapy achieved remission. People think of BDD as an incredibly severe disorder, and it really can be. So these are promising data that show that in both conditions, especially after CBT, remission is likely. And on the right, we show remission-related outcomes at 6-month follow-up. And so I'll just highlight two. In blue, we show the proportion of participants in each condition who remained in remission from end of treatment to 6-month follow-up. And in red, we show those participants who never achieved remission at either post-treatment or 6-month follow-up. So again, you can see how those outcomes, they vary by condition, and overall we're really quite strong 6 months after a course of CBT for BDD.
Thank you so much, Hillary. Sabina, next, Olivia.
Hello, my name is Olivia Okereke. For over 15 years, I've led a research program on finding paths to better mental aging. The main areas are preventing depression, preserving cognition, and avoiding functional impairments. We use methods in epidemiology and prevention science and advanced statistical techniques with large data sets of well-characterized participants. In one study of over 20,000 people, we identified risk factors for late life depression. Modifiable factors such as avoiding smoking and heavy drinking, getting more exercise, better sleep, and healthier diet, and maintaining a larger social network were related to lower risk. We estimated 1/3 of all cases of late life depression might be prevented by changing just 3 things: mitigating bodily pain, increasing exercise, and addressing poor sleep. Our epidemiologic work informed the measures we collected to assess risk mediators and moderators in a recent large depression prevention trial. We tested prevention of depression with 5 years of randomized vitamin D and omega-3 supplements in 20,000 diverse older Americans. We did detailed assessments in over 1,000 of them who were local to the Clinical Research Center in Boston. I'll highlight some key findings. While vitamin D did not benefit mood or prevent cognitive decline in the full sample, there were significant benefit among Black African American adults for preventing cognitive decline. And this was notable given the higher vulnerability of Black adults to low vitamin D status. While there was no overall benefit of 1 gram a day of omega-3, there were significant sex differences in response with regard to mood, with some indication that hormone status in women contributed to the variation. We recently received new NIH funding to probe the biological mechanisms of these effects using novel blood genetic and imaging measures. I hope this brief presentation gives you a sense of our priorities and activities. Thank you for listening.
Oh, thank you so much, Olivia. Next, Ana Maria.
Good morning, everyone. We are the Center for Health Outcomes and Interdisciplinary Research or CHOIR. We focus on developing, testing, and implementing technology-enhanced mind, body, and lifestyle interventions for patients, caregivers, and dyads of patients and caregivers. These interventions are focused on preventing illness, optimizing recovery after illness, and promoting living well with persistent or serious illness. We are a clinical research and training program. All team members of CHOIRs have been clinical psychology interns or postdoctoral fellows. We work with a variety of medical populations within MGH and alongside national collaborators, with more recent work moving into community settings and underserved clinics. Next slide, please. So our process of intervention development through implementation is guided by the NIH stage model, and the figure on the slide exemplifies this process for one of our current projects. So the aim of the Get Active Fitbit study was to develop a mind-body activity program for people with heterogeneous chronic pain through combining mind-body skills with gradual increase in step count linked to valued activities. With an NCCI-HR 34, we developed an iteratively refined intervention using qualitative and mixed methods. We use this information to secure an NCCI-HR 01 to determine multi-site feasibility of the programs at 3 geographically diverse pain clinics at MGH, Duke, and Rush Medical Center. And Dr. Jonathan Greenberg, who is a postdoc on R34, joined me as NPI on this project. We will next apply for a multi-site hybrid efficacy effectiveness trial, which we hope will support implementation of Get Active Fitbit in the routine care of people with chronic pain within pain clinics across US. I'll stop here.
Thank you so much, Ana Maria.
Hi, my name is Abigail Batchelder, and I am a clinical psychologist in the behavioral medicine program. And I'm going to tell you a bit about the stigma and substance use research being done in the behavioral medicine program. Our aim in this work is to expand the BMed research portfolio to include studies focused on understanding and intervening on the impact of intersectional stigmas affecting health behaviors, with a particular focus on marginalized people, including sexual and gender minority individuals, and those living with HIV and/or substance use disorders. We have a range of projects underway currently, including work with individuals living with HIV who inject drugs, and men who have sex with men living with HIV and active substance use disorders. Those are both stigma reduction interventions or interventions to address the impact of stigma on health. I'm also doing some measurement work looking at emotions to inform intervention strategies, as well as investigating sexual minority folks' engagement in recovery community centers to inform interventions, and expanding some of my clinical work with people aging with HIV. Some recent exciting findings involve the identification of the perpetuation or the bidirectional relationships between negative subconscious emotions, stigma-related emotions, shame and guilt in particular, and substance use, identifying that shame and guilt are barriers to the reduction of stimulant use in particular. Some of my recent work has also identified substance use internalized stigma in particular, as a barrier to engagement in HIV self-care among men who have sex with men, identifying it above and beyond the barriers imposed by HIV and sexual orientation related stigma. So really making it a prime target for intervention strategies. Through this work, I would welcome collaborations, and thank you so much for including me.
I'm Dr. Gagan Joshi. Thank you for the opportunity to share our latest research findings from Bressler program for autism here at MGH. At Bressler program, we conduct studies to study the clinical and neural correlates of autism spectrum disorder and frequently associated psychopathologies, with the aim to improve their diagnosis and their management. I'll be discussing 2 sets of studies. In the first set of studies, we conducted a neuroimaging study to study the neurotransmitter glutamate activity in a particular region of the brain, which is responsible for social emotional functioning. The region is called pregenial ACC. And what we found that in a subset of individuals with autism, they have an abnormally high glutamate activity in the pregenial ACC. With the information that the glutamate activity is abnormal in a subset of individuals with autism, we conducted a clinical trial with a glutamate modulating agent called memantine to see if it can improve social emotional functioning in individuals with autism. Our findings suggest that memantine is safe and well tolerated. And in addition, memantine had a significant response in improving social emotional functioning only in those individuals who had high glutamate activity in the pregenial ACC. So it appears that we have a biomarker for predicting response to glutamate modulating agents in autism population. In our second set of study, we looked into the ADHD comorbidity with autism spectrum disorder. And what we know that ADHD is the most frequent psychiatric condition associated with autism and has excellent treatment if identified in this population. For historical reasons and for some diagnostic reasons, ADHD is underdiagnosed in autism spectrum disorder population. So we embarked into a two set of studies to look into the clinical and the neural profile of ADHD in autism population to see if it expresses itself in autism population the same way as it does in the typical population. In the first table you see here in this graph, in the orange are the ASD individual with ADHD symptoms, and in the blue are ADHD without ASD. And what we notice that the expression of ADHD in the autism population is very similar to what you see in the typical population. So expression of ADHD is not atypical in ASD population, and so can be easily clinically identified using the same parameters what we use in the typical population to recognize ADHD. Now, the second set of study we did, we looked into the neural profile of ADHD in the individuals with autism, who had also ADHD clinical presentation. And what we found here, that the structural connectivity, there was underconnectivity of the major tracts in the brain called the cingulum corpus callosal tract, where this underconnectivity, what was observed with ADHD, equally expressed in the ASD individuals who clinically presented with ADHD. So taken together, it tells us that the ADHD clinical presentation as well as neural presentation is similar in autism individuals, what you see in the typical population. And so they would also respond to the treatments what are offered for the typical population. With this, I would say thank you for the time and interest and for your support. Thank you.
I'm Gary Strangman, Director of the MGH Psychiatry's Neural Systems Group. Our mission is to help improve the mental health and performance in real-world settings by pushing the frontiers of brain assessments and interventions beyond the hospital walls. We specifically focus on applications in extreme and remote environments, which inspires our mobile platform development. The domains that we work in are listed here, ranging from spaceflight to first responders to a wide range of patient populations. Prior to 2020, our group consisted of just 3 investigators, and since then we have added 5 new staff members as well as a dozen interns. Our biggest recent achievement is a major expansion of our space medicine research program. Since early 2020, we have grown from 6 projects totaling $3.5 million to 15 projects and $8.2 million in research funds. New projects include developing a closed-loop biofeedback platform to optimize individual performance, studying sensory motor performance in partial gravity conditions, identifying team performance challenges in operational settings, assessing astronaut sensory motor capabilities on landing day to guide future Mars missions, investigating the relationship between microgravity, immune function, and emotion perception, and testing virtual reality as a countermeasure for isolation and confinement. There wasn't room on the slide, but other new projects are examining electrical brain stimulation to combat the stresses of spaceflight and long-term astronaut brain health monitoring. Importantly, this work also has numerous Earth-relevant applications by enabling neurophysiological monitoring at home, in office or clinic settings, in perioperative settings, and in real-world settings such as exposure therapy and first responder training.
Hello, my name is Dr. Oliver Freudenreich. I'm the co-director of the MGH Psychosis Program. And I would like to introduce you to a smaller service within this larger program, the MGH Treatment-Resistant Schizophrenia Service. I am the director of the service with a mission to improve the care of patients with treatment-resistant schizophrenia. And in order to achieve this mission, I delineated 4 goals. We want to innovate, meaning we want to develop more effective and safer treatments. We want to integrate, to have fewer silos and more partners. This is actually very important. The integration is not just within our own department, but within MGH and different departments like neurology and medicine, but also within the larger community. And we do work collaboratively with the Department of Mental Health, for example, or our longstanding community partner, North Suffolk Mental Health Association. We want to instruct, I want to provide easy access to consultation for colleagues and families. And last, overarching, perhaps we want to influence the care of patients with treatment-resistant schizophrenia by disseminating knowledge. And in order to achieve these goals, we have 4 separate yet interdependent clinics or groups. We have the clozapine clinic, which is directed by Carol Lim. We have the clinical trials group working with industry to develop, bring to market new treatments. My longstanding nurse, Sarah McLaurin, is in charge of the Med-Psych integration working group. And last, we have a very active psychosis consultation service. Very pleased that I can give you an example that was just published about recent work. It was published in Psychiatric Services, our vaccine project. It's a great example of our attempt to integrate medicine and psychiatry, ultimately in this case to reduce mortality for patients with treatment-resistant schizophrenia. And we did this with the help of our community partner. Here on the right, you see the Eric Lindemann Mental Health Center, where the Friedenfeld Clinic is located, which is one of the community sites for our MGH North Suffolk clozapine clinic collaboration. When the pandemic in early 2020 hit us, it became fairly clear quickly that patients with serious mental illness are at high risk for COVID-19. Schizophrenia actually was shown to be the largest predictor of mortality from COVID-19 after age, and somewhat related, this patient group has remained under-vaccinated despite vaccine availability with, for example, the flu vaccine. So when the vaccines became available, we really thought that we as psychiatric providers are well positioned for targeted outreach to improve the vaccination rates for patients with serious mental illness. We are often their primary point of healthcare, and we are experts as psychiatrists in behavioral management techniques such as motivational interviewing and nudging. So we took this clozapine cohort that we have in the Lindemann, about 200 patients, most with schizophrenia, across a broad range of ages here. And we really only did 2 interventions. We provided in-service education to all the psychiatric providers, give them strategies to address vaccine-related concerns and overcome practical barriers to vaccinations. And then we embedded a vaccination monitoring tool in the electronic medical records, tracking vaccination intention, hesitancy, and uptake at each visit to give us a framework to follow up and really manage this population as a whole population. And I'm very proud to say, maybe this is one of my proudest moments in my last 20 plus years working in community psychiatry, that we achieved significantly higher vaccination rates than the rate of statewide vaccination in Massachusetts. In our cohort in this period from February to June of 2021, we reached 84%. Whereas the adults in Massachusetts had a rate, depending how you measure it, between 62 and 77%. So this is one example that also led us now to really get more involved in public health efforts and more broadly in vaccine preventable illnesses beyond COVID. One example again of one of the things that we're interested in serious mental illness, improving mortality through better integration between medicine and psychiatry, but also between MGH and the community. Thank you.